So let's get started. So hopefully everybody was able to watch the chapter 1 lectures and maybe do some of the posted practice questions and look through the answers. So today's session, we're going to go over some of those additional review questions that are part of the slides from chapter 1. So hopefully we can have a good back and forth. I don't want to do the problems for you. I want you to tell me how to do them, help each other out, that kind of thing. So just as a review, this chapter covered 3 different categories or topics. The first topic was origin of life and functional groups, so we'll do a few review questions about that. Then we'll have some review questions about the cellular architecture portion, and then finally we'll do some review questions about thermodynamics. So we'll see, hopefully everybody was able to understand, and if not, that's what this is for, to help everyone make sure we all know what's going on. So first we'll start with the origin of life review questions. So can anybody tell me the monomeric building blocks that make up DNA and RNA? Yeah. Nucleotides, yes. I'm sorry? No, I'm sorry. Yeah, so DNA and RNA are made from nucleotides, so they are polynucleotides or nucleic acids. Can anybody tell me the building blocks of proteins? Yeah, amino acids. So amino acids are made into peptides, and long peptides are known as proteins. And finally, can anybody tell me the building blocks that make up carbohydrates? Yeah. Monosaccharides, yes. So monosaccharides are made into polysaccharides, which form complex carbohydrates. All right, hopefully that's pretty straightforward for everyone. So the next question, what type of reaction are we looking at here? And there are 5 choices for you to pick from. Yeah. Condensation, yes. This is a condensation reaction. So we're bringing together 2 monomers to make a polymer, and we're removing the OH from one and the H from the other in the form of water. So this, can anybody tell me what this functional group is that I just circled? Yeah. Carboxylic acid. And what about this one? Yeah. Amine. Can anybody tell me what type of bond it is when you bring together a carboxylic acid and an amine? Yeah. It's an amide bond. Yeah, and in this case, these are 2 amino acids, and when you bring them together, it's specifically called a peptide bond, which we'll go more into later. And how do we, I guess I gave away that these are amino acids, but the way we know these are amino acids is, hold on, let me erase this. So amino acids have an amine on one side, a carboxylic acid on the other side, and then there's a central carbon that has an R group. And we will learn about the 20 most important of these when we cover amino acids. So just one more question about this. If I was going to do the reverse reaction, if I wanted to add a water to break apart this larger molecule, what would that be called? Yeah, hydrolysis. Excellent. All right, great. So now we're going to look at remdesivir, which may or may not sound familiar. It's an antiviral drug that people were hopeful would be useful against COVID-19. It turns out it's not super effective, but it is effective against a number of other viruses. So for this question, I'm going to ask if anybody can identify the functional groups. So this one on the left, this whole thing, can anybody tell me what that is? Yeah. An ester. And just as a review, can anybody tell me what just the C double bonded to O is called? Yeah. Carbonyl. All right, so this next one that I'm circling is maybe a little bit less familiar, but we did go over it in the lecture, and I believe even last week during the review. So can anybody tell me what that functional group is called? Yeah.
Phosphate diester?
Yeah, excellent. So that's a phosphate diester. So phosphate diesters, they have a central phosphate that's bonded to 3 oxygens. And those 2 of the oxygens, you sort of have 2 esters. That's how you can see. So you make 2 linkages. So you have one kind of phosphate ester here. You have your second phosphate ester there. Sorry, that's extremely messy. So you have this sort of forms one phosphate ester that's linking to this other piece. This is sort of forming a second ester. That's why it's called like a phosphate diester or a phosphodiester linkage. It's linking together 2 R groups. Okay, so can anybody tell me what, just erasing this so it's a little less messy. Can anybody tell me what this functional group is called? Yeah, ether, great. And how about this one on the bottom? Hydroxyl, excellent. And hydroxyls are sometimes also called alcohols. And all right, so finally in the upper right corner, what's that one? Yeah, amine, great. Okay, great. So looks like y'all remember your functional groups. Sure. Oh yeah, so this doesn't have to be a 4th oxygen. It can be something else. The important thing for a phosphate diester is you have like the central phosphate that's double bonded to an oxygen, and then that second oxygen, it looks kind of like a regular ester but with a phosphate as the central molecule, and you have 2 of them, sort of one on this side and one on that side, if that helps you identify it. Yeah, so this doesn't have to be an oxygen, and in this case it's a nitrogen. Yeah. Yes. Mm-hmm. Yeah. Yes. Mm-hmm.
So then to add on to that, so
Yeah.
the phosphate ester is only different than OH instead of the NH.
So a phosphate ester. So that's just a single ester. Yeah, so that would be, yeah, so if you removed the rest of this molecule and this was just an OH and it terminated there, then you would have just one ester linkage. The other one is sort of more like an acid. It's terminal. So a phosphate diester has sort of 2 R groups. So you have P, O, 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 R, R. There's 2 R groups. And then this can also be an oxygen or whatever else. Yeah. Yeah.
Swap that amine attached to phosphate for another oxygen.
Mm-hmm.
Would you have a phosphate triester?
Like if this oxygen was also attached to an R group?
Yeah.
Yeah, I guess so. I've never seen that biologically. I don't know if that can happen, but I guess it could. Yeah. Yeah. Yeah, so the functional group is called a phosphate diester sort of on its own, but the linkage sort of refers to the fact that it's linking together other pieces of the molecule. So it's sort of like with hydroxyl, like this is a hydroxyl group, but like the molecule altogether is an alcohol. It's sort of 2 terms. So like the linkage sort of refers to what it's doing, like its function in a way. Yeah, you could call it either one. I would consider, if you said this is a phosphodiester linkage, or if you said this is a phosphate diester, I would say that's correct. Kind of like if you said this is an alcohol group or this is a hydroxyl group, I would say that's fine. Yeah. Any other questions? Yeah.
The list of functional groups
Yeah.
on the slides, I saw there was an amine. Do we not see enamines in bio?
They're not very common in biological macromolecules. Yeah, that list is really just like the ones you're going to see most frequently. So you learned probably about a bunch of other ones through organic chemistry that are very important in their own right, but yeah, they're not very commonly seen sort of in the stuff at least that we'll be looking at in this class. Yeah, I'm trying to think of, there might be like some cases where you might see one, but it's not worth getting into right now. Yeah.
Does a phosphate ester, when it's like on its own, have to be bonded to an OH or is it just the fact that it's like phosphorus double bonds to oxygen and then single bonded to another oxygen?
So yeah, so a phosphate ester, just one ester, instead of that second R group, you would have the OH. Or I think one of these is actually a double bond, but yeah, I can't count. I can never count these. Sorry. Okay, so yeah, so a phosphate ester has just one R group. Phosphate diester has two R groups.
So the minute the charge like oxygen could be literally anything else, but the rest of the molecule is what?
Yeah, so you can think of a phosphate group as like, this is phosphate, PO3, like that's phosphate. Like if you want to stick other stuff onto it, that's where you start getting into, it's like an ester. So kind of like a thiol is like an alcohol but with a sulfur, a phosphate ester is like an ester but with a central phosphorus, if that helps.
Yeah.
Okay. Yeah. Yeah.
Doesn't that reduce the evolution of Linus model instead of kind of like changes depending on if it's the phosphate ester or the diphosphate? The like
Yeah,
all Linuses will move. Does that matter when you're drawing it?
No. Yeah, it's just sort of however you like to draw it. They can move around in space, so it's sort of, you can think of it as like that's the snapshot when you drew it, but they can move around.
Okay.
Yeah. All right, if there are no more questions, so now that I scribbled on this, there's a little bit, if you're interested in remdesivir itself as a drug, it acts to inhibit RNA polymerases, so it stops viruses from being able to replicate their genome and proliferate by basically clogging, that's the wrong word, by causing like a chain termination or a stalling in the RNA polymerase as it's trying to do its job. So if you're interested in learning more about exactly how that works, there's this nice little article here. Okay, so that's the end of our sort of functional group review. If you have any other questions about that, please come to my office hours, email me, etc. So let's move on to cellular architecture. So for each one of these statements, I want to know, does it apply to prokaryotes, eukaryotes, both, or neither? So number one, which ones typically reproduce on the timescale of minutes? Yeah, prokaryotes. Yeah, excellent. Yeah, prokaryotes tend to replicate much faster. Eukaryotes tend to replicate a little slower. One of the reasons for that, to maybe help you remember, eukaryotes are kind of a little bit more complicated, right? They have all these organelles inside. There's a lot more stuff going on in a eukaryotic cell. So you can think of it as sort of being almost like a little bit more work. So it might take them longer to replicate. Okay, so which ones can be multicellular? Yeah. Prokaryotes? Eukaryotes, yes, excellent. Sorry, I have bad hearing. Excellent, so eukaryotes. both eukaryotes and prokaryotes can be unicellular, but only eukaryotes can also be multicellular. So which one has smaller cells on average? Yeah, prokaryotes. Yeah, if you want to see them in a microscope, you need really, really high magnification, much higher than you would need to see eukaryotic cells. They're really tiny. So, okay, so which ones have ribosomes? Both, yes. So both prokaryotes and eukaryotes need to be able to make proteins to do their jobs, and to do that, they both need ribosomes, because ribosomes help make proteins out of an RNA template. Okay, so which ones have a nucleus? Yeah, eukaryotes, excellent. So to add on to that, which ones have DNA? Yeah, they both have DNA. Both of them tend to put that DNA in like one part of the cell, but only eukaryotes have a nucleus that has a membrane that more completely surrounds and protects the DNA. Okay, so which ones have a cell membrane? Yeah, both. Yeah, so a cell membrane kind of defines where is the cell, what's inside the cell, what's not in the cell. If you are a cell, you have a cell membrane, whether you're a prokaryote or a eukaryote. Okay, does anybody have, yeah?
What are like the membrane found organelles that it's lacking in the prokaryote?
So prokaryotes, the one membrane-bound thing that they have is just the cell membrane that surrounds the whole cell. They don't have any internal organelles within the cell that are surrounded by membranes. So they might have areas, like there's an area usually of the cell where all the DNA is, but it's not inside of a nucleus, right? It's just kind of there in its own space. So what defines eukaryotes is the membranes that define each organelle. That gives it a compartment with a function. All right. Any other questions? Okay, so now let's move back toward talking about those eukaryotic cells. They have a number of different organelles that we're going to be talking about, and they each tend to be locations where different processes occur. So who can tell me which organelle where DNA is stored? Number one. We just talked about this. Yeah, the nucleus. Where is DNA synthesized? Yeah, also in the nucleus. Does anyone remember what that process is called when DNA is synthesized? Yeah. So translation is a couple steps ahead. That's making proteins. Yeah. Replication, yeah. So DNA is replicated. DNA uses, to replicate DNA, you have DNA itself that you're using as a template that you're making a copy of. You're replicating it. And that happens in the nucleus because that's where the DNA is and you need to use it as your template. So question number 3, synthesis of RNA. Where might that happen? Yeah. So that's also exactly right. Yes. So that's exactly right. So when you make RNA, you're also using DNA as a template and you're copying the RNA off of that. That's going to happen in the nucleus. After the RNA is made, like you said, it then moves into the cytosol to get made into protein.
I might be remembering this wrong, but Isn't RNA like also like more specifically in the nucleoid region? Is that kind of different?
Are you thinking of the nucleolus or?
Maybe.
So I think I'm trying to remember. Yeah, so the nucleolus is where, now I'm blanking on what that happens, but that is, I'm pretty sure that's where ribosomal RNA is made specifically. I will double check on that and get back to you because I just blanked on it. But does anybody remember, to get back to this question, about the synthesis of RNA? What is that process called? Yeah. Transcription, yes. Cool. Okay, so now we're sort of going in order. So we made this RNA now. We're going to try to make that into protein. So question 4, if you're making a cytosolic protein whose end location of where it's going to live is in the cytosol, where is that going to happen? Yeah. I'm sorry, I didn't. So yeah, so for proteins that are just going to live in the cytosol, all they actually need are free ribosomes that are in the cytosol also. So, but you're kind of bringing us towards the next question about synthesis of secreted proteins. So the cell separates where it makes cytosolic proteins that are going to live in the cytosol and where it makes proteins that are going to live inside of a membrane or get secreted. So where in the cell are the secreted and membrane-bound proteins made? Yeah, the rough ER. Yeah, exactly. And when the cell is making proteins that are for secretion in the rough ER, it goes through a special process that basically helps direct them where to go. If it made them just within the cytosol, they would be kind of stuck there, and they couldn't get then directed further. All right, so number 6, modification and packaging of proteins. Yeah, the Golgi, excellent. So, whoops. Yeah, so for proteins that are going to get secreted or for whatever reason need extra pieces onto them after they're made in the ER, they get sent to the Golgi. The Golgi does further modification and tells them where they're going to go. All right, so last we have number 7, the synthesis of ATP by ATP synthase. Where does that happen? Yeah, mitochondria, great. All right, does anybody have any questions about the organelles, cellular processes, anything like that? Okay, so, all right, now I'm going to ask you to correct each one of these statements. So the first statement says, the individual cells within a human are prokaryotic, while the entire human organism is eukaryotic. So can someone tell me either what's wrong with that and also how they might fix it? Yeah.
Well, I mean, for starters, you're not going to have one organism that is both eukaryotic and prokaryotic.
Mm-hmm.
It's confusing, eukaryotic doesn't have one cell, whereas prokaryotic will move the whole cell.
Right. Okay. So within a human, is it correct to say the cells within a human are prokaryotic? No. And you'd fix that by making them eukaryotic. Yeah, exactly. All right, so the second one says, a homogeneous population will be better able to respond to a new pathogen because all individuals have evolved to have strong defenses against infection. So this is a little bit part of that evolution stuff that was in the lecture. So is this right or wrong? We already sort of know it's wrong. So what is wrong and how might you fix it? Yeah.
Folks could actually be more susceptible because since there are many genes, they all have the same genes, and since it's a new pathogen, if they get infected, they're all going to get infected, essentially they'll die.
Yeah, exactly. So a homogeneous population, if they're susceptible to one thing, everyone is susceptible to it. So that's a problem. So another way that you could fix it maybe is by changing it from homogeneous to heterogeneous. So this is a population where there's a little bit more genetic diversity. And can anybody tell me why that type of population might be better able to respond to a new pathogen? Yeah.
You have like more genetic diversity, so then those genes will be able to better adapt to changes, and then those with like the best traits will produce more offspring that have good genes.
Yeah, exactly. So like with a heterogeneous population, some people might be susceptible, some individuals might not be. So those individuals would be able to survive the pathogen, and those individuals, like you said, would then be able to survive and reproduce. Great. Okay, so number 3. When a population of bacteria are exposed to antibiotics, some of the bacteria mutate to become resistant to the antibiotics, and that allows them to survive and replicate. So you already know that this is incorrect because I'm asking you to correct it. So yeah, can anybody tell me maybe why this is wrong and how they would fix it? Yeah, go ahead, sorry.
It's not that they mutate, it's that they are able to survive and therefore they're able to replicate. It's not, they don't mutate in their
Yeah, that's a really good start. So it's like you said, they're not mutating in response to the antibiotic. It's sort of like building on the last question. They're a little bit of a heterogeneous population. So as bacteria are replicating, some of them will have random mutations that will slightly alter some of them. So some of them may be more susceptible or less susceptible to something like an antibiotic. So the ones that are more susceptible will not survive being exposed to an antibiotic. The ones that are more resistant or less susceptible naturally will survive being exposed to the antibiotic, and those bacteria will be then selected, and those bacteria will survive and replicate. So antibiotics help to select for already existing resistant bacteria. Does that make sense to everyone? Okay. So let's see how we fix this. Some of the bacteria are already resistant. So that's maybe the quickest way to fix that. But if it was on a test, maybe you could add a little bit more to that. Okay, so does anybody have questions about any of these questions that we just went over? All right, that brings us to thermochemistry. So we're going to do some math now. So I guess before we start, just in general, I just wanted to say there's going to be math on your quizzes, on your exams. For just practice questions or even for the quizzes, you're welcome to use your phone, a tablet, whatever has a calculator on it that you like. But for the exams, I'm only allowing just regular calculators that are only calculators, not phones, not tablets, no internet-connected devices. So I would recommend doing practice problems on the device you're going to use for the exam, just so you're not like panicking on exam day. But that is up to you. So that being said, did you have a question before we start? Or?
Yes, I have.
Okay.
Tracking calculators, okay?
Yeah. Yes, I guess as long as they're not, internet connected or nothing crazy's going on. Okay. Okay, great. So we're going to start by talking a little bit about Gibbs free energy. So this question says, consider the reaction A is made into 2B at 298 degrees Kelvin, for which the change in enthalpy is 22 kilojoules per mole, and the change in entropy is 193 joules per moles Kelvin. So the first part of this question, is this reaction spontaneous under standard conditions? So the first question, so it's not, you can't tell this just by looking at it, right? You have to do some math to figure that out. So can anybody tell me what equation they might want to use to figure out if the reaction is spontaneous? Yeah. Yeah, exactly. So delta G equals delta H minus T delta S. So which variable in there is going to tell us whether or not the reaction is spontaneous? What do we need to solve for? Delta G, yeah. So delta G. Okay, so who can help me fill in this equation? So for delta H, can I just fill in 22 kilojoules per mole? Is that cool with everyone? Okay. All right, what do I fill in for T? Yeah, so it's under standard conditions, so that's 25 degrees C, but we always have to do everything in Kelvin, right? So 298 degrees Kelvin. Or if you want to be very exact, you could do 298.15. Either way is fine. And then what do we fill in for our delta S? Yeah. Yeah, excellent.
It should be 193 times 1 times 10 to the minus 3.
Mm-hmm.
Times.
Yeah, exactly. So there's 1,000 joules in a kilojoule. We divide joules by 1,000 to get kilojoules. So another way you could do that, if you ever forget if you're supposed to divide or multiply. So if you have joules, you want your joules to cancel, so you get kilojoules. There's 1 kilojoule and 10 to the 3rd joules, like you just said. So 193 divided by 10 to the minus 3. Move the decimal place over by 3, so it's 0.193, and that's kilojoules per moles Kelvin. Okay, so that's, now we have our delta S term all ready to go. So we have 0.193 kJ per moles K. All right. Does anybody have a number for what is delta G? Fabulous, yeah. So we have negative 35.5 kilojoules per mole. And in general, when I ask you for delta G, it will almost pretty much always be in kilojoules per mole. So, yeah, you can, the other way you could have maybe done this is by changing the kilojoules per mole in your H term to joules per mole, but then if you did that, everything would be in joules per mole at the end, and you'd have to switch back to kilojoules. So this way is sort of faster, because you're starting out with the units that you want to end up with. So now we can finally answer this question, right? Is the reaction spontaneous? Seeing some nodding. So yes, the reaction is spontaneous. Why is the reaction spontaneous? Yeah. Delta G is negative. Yes. So if delta G is less than 0, we are spontaneous. Cool. So let's okay, so I guess to follow up with that question, given this reaction, what could you do to make this reaction non-spontaneous? What could you change about it? Yeah. So exactly. So you'd have to change it from being standard conditions to non-standard conditions, which would affect, so you can change the temperature. So in this case, because you have a positive delta H, if you have a low value for the temperature, then you could make this term basically something less that you're subtracting, so you'd end up with a positive delta G, if that kind of helps to visualize it. Okay, so part B, so same reaction, same delta H, same delta S. So at what temperatures will this reaction be spontaneous? So we were just saying how at some temperatures it's non-spontaneous, at some temperatures it is spontaneous. So how can we figure out where the dividing line is? Yeah. Yeah, exactly. So delta G equals 0 is sort of that dividing line, because if delta G is higher than 0, it's not spontaneous. If delta G is less than 0, it is spontaneous. So if you figure out the temperature where delta G is 0, that's right in between. And then we can figure out which ones are above, if it's above or below where it's going to be spontaneous. So if we set delta G equals 0, we can use our same Gibbs free energy equation. So delta G equals delta H minus T delta S. And now we're setting delta G to 0, so 0 equals delta H minus T delta S. So we can add T delta S to both sides, and so you end up having T delta S equals delta H. And we want to solve for temperature, right? So we divide each side by delta S, so then we get T equals delta H divided by delta S. Does that make sense for everyone? So now we can figure out what the temperature is where delta G equals 0. So what would I fill in here for delta H and delta S? Yeah, exactly. So we use our same delta H from before, so 22 kilojoules per mole. And again, we have to convert our delta S to kilojoules per mole, which we already know is 0.193 kilojoule per mole. Okay, so now with temperature, these cancel out, we're left with temperature, so 22 divided by 0.193, like you said, is about 114. And so now, did you tell me if temperature is over or under 114 are the spontaneous ones? Yeah, so if you're ever not sure, a good way to do it is to test it out using our original equation. So we could plug in, so we actually already did this in our last problem, right? We did this at standard conditions at 298 K. 298 is above 114, right? And we already know it's spontaneous at that temperature. So now, and we also know now that 114 is our dividing line. So anything above 114 is going to be spontaneous. Does that make sense for everyone? Okay. Okay. So now our last question is, what will happen to the spontaneity of the reaction? Or did you have a question? Yeah. Yeah.
I was having a little brain fart. Can you just go over the breakdown of the Brownian equation within TM on the side?
Oh, absolutely. Yeah. So let's start again. If I can erase this. Okay, so we have our Gibbs free energy equation, delta G equals delta H minus T delta S. And we know we want to set our delta G to 0 because we want to find we're looking for what's happening at equilibrium. Does that make sense? that you want to find when g equals 0? Okay. So if we set g equals 0, now we can change this delta g term to a 0. So now we have 0 equals delta h minus t delta s. So now from this point, we're just trying to isolate t because we're trying to solve for t. So I'm going to add t delta s to both sides of my equation. Now on the left side, I have t delta s. On the right side, I have delta h. And then to isolate t, we divide both sides by delta s. So then the delta s's cancel out. On the left side, we just have t. On the right side, we have delta h divided by delta s. Is that good? Okay, cool. And you got the rest made sense, right?
Yeah.
Okay, great, cool. Okay, are there any more questions about this part? Okay, so the next part of the question is, what will happen to the spontaneity of this reaction if we increase the temperature? Yeah.
Is it more spontaneous?
Yeah, basically. So the more you increase the temperature, the more negative delta G gets, like we were saying, which sort of makes it more spontaneous. What does that mean for the reaction? Like, is it going faster or slower? Mm-hmm. Yeah. No. So, sorry. But the spontaneity of a reaction only tells us whether or not it's going to go. It never tells us how fast or slow it's going to go. All delta G tells you is this reaction is favorable and it will eventually happen. So it doesn't matter how negative it is. But the more negative it is, it tells you the balance of where the reaction will go, where it's at equilibrium. So if you have a more spontaneous reaction, when it finally does reach equilibrium, you'll have more products. If you have a reaction that's spontaneous but like less spontaneous, you still have more products than reactants at the end, but the ratio of products to reactants is sort of closer to 1. Does that kind of make sense a little bit? Yes, no? Okay. Any questions about that part? Okay. So, another Gibbs for energy type question, but now we're going to focus more on the equilibrium constant, which kind of ties us back in from that last question. So, new reaction, 2A makes B plus C. We're at 298 K. When this reaction is at equilibrium, we have the following concentrations of A, B, and C. I will not read them out loud, but they're right on the slide for you. So first part of the question, what is the equilibrium constant expression for this reaction? So equilibrium constant is K. So when I ask you for the expression, I'm asking you to tell me not with numbers, but with letters, sort of giving me or using the information from the equation. Yeah. Yeah, exactly. So K, just to review, we have we have products, concentration of products divided by concentration of reactants. My pen is going a little wild. It says reactant. And just like you said, so our products are B and C. So concentration of B times concentration of C. Our reactants go in the denominator. And that's 2A. So for when you're calculating K, you use the power of the coefficient up here, so it's A squared. If you just had 1A, it would just be concentration of A. If you had 3A, it would be A cubed. So, any questions about the expression, the equilibrium constant expression? All right, so now that we figured out the expression for K, let's figure out the numerical value of K. So what would I fill in there? So we have concentration of B, concentration of C, divided by concentration of A, squared. So sort of, I guess this is pretty straightforward. So concentration of B, right? We have 0.0028 molar. Concentration of C, we have 0.0073 molar over A squared, and A is 0.019 molar squared. So, yeah. Yeah, so it's because when you look at the chemical equation, you have 2A. So 2, the coefficient in front of A is going to be the power when you're making the equilibrium constant. So if this said 3A instead, then the denominator in the equilibrium constant would be A cubed. Or if it was just 1A, it would be just A. So does anybody have a number for the equilibrium constant? I'll give you a sec to calculate it out. I'm sorry, I didn't hear you. Yes. So what units is the equilibrium constant in? No units. Yeah, the equilibrium constant is always unitless. There are never any units. And in this case, you can even see that the units are going to cancel out, right? You have molar, you have molar times molar on top, and you have molar squared on the bottom. So the 2 molars on top cancel with the 2 molars on the bottom. You have no units. You just have numbers. The other thing I want to warn you about whenever you're calculating K, always make sure that the numbers you plug in are in molar, which is moles per liter. If I give you it in millimolar, which I might sometimes do, first change it to molar before you do the math. Otherwise, you will get the wrong equilibrium constant. Does anybody have any questions about this part? Okay. So, next part. So same reaction. What is the standard free energy change, delta G naught, for this reaction? So first of all, what is the equation, does anybody remember, that we use to calculate delta G naught? Yeah. Yeah, except it's negative, but yes. So delta, oops. Sorry, why is this happening? Delta G naught equals negative RT times the natural log of K. All right, so do we have everything we need to start solving this equation? So we have the temperature, right? That's T, 298 K. We have K, the equilibrium constant, because we solved for that on the last slide. So K is 0.057. And then we need R. So where do we get R from? R is a constant. Yeah, so R equals 8.314 joules per mole Kelvin. And that's something that you'll always be given on quizzes and exams. You don't have to remember the value of R. But we do always have to watch out for units. So remember, delta G is almost always reported in kilojoules. This R value, it's got joules in it. So to make our lives easier, we might sometimes want to convert that to kilojoules per mole K instead. So, okay, so let's plug everything in. So delta G naught equals minus, so R, so we can we can leave it the way it is if we want, 8.314 joules per mole Kelvin times temperature, that's 298. Okay. And then the natural log of the equilibrium constant, so that's 0.057. Does anybody have, so that's one way you could solve it. The other way you could solve it, or I guess let me just say it, so that will end up giving us, the K's cancel out, so you end up with something joules per mole. You have to change that to kilojoules per mole because we're reporting delta G. So the other way you could solve it, so you end up with kilojoules per mole, is you change R. So instead of making it 8.314 joules per mole K, you can divide by 1,000 to get yourself kilojoules. You'd have 0.008314 kilojoules per mole. Okay. So if you do it that way, 0.08314 joules mole Kelvin times 298 K times the natural log of 0.057. So this one, if you solve it that way, you'll end up with your final answer and you don't have to convert anything. Either way is correct. So what answer do we get? I'll let everybody do the math for a second. Yeah, exactly. So you have 7.1 kilojoules per mole. If you did it the other way, where you got joules first, you would end up with 7,000 and about 114 joules per mole, which you would then divide by 1,000 to get your kilojoules per mole, because this is your final answer, 7.1 kilojoules per mole. Does that make sense to everyone so far? Okay. So something else we can start thinking about with the equilibrium constant is, so that kind of brings us back to this. So the equilibrium constant is always the concentration of your products over the concentration of your reactants. And you can sort of start linking that to whether or not a reaction is spontaneous. So a spontaneous reactant is going to lean towards products at equilibrium. So a spontaneous reaction is going to have a K that's greater than 1 because the ratio of products to reactants, there's more products, so the ratio is greater than 1. If you have a non-spontaneous reaction, it's going to lean towards reactants. It's not going to want to go to product. A non-spontaneous reaction will have a larger denominator than the numerator. You'll have a K that's less than 1. That's something that you can keep in mind. That's sort of maybe like a check as you go through these problems. If my reaction is spontaneous, I really hope my K is greater than 1.
Could you repeat that? If it's non-spontaneous, it leads towards the reactants?
Yep. Mm-hmm.
And then K will be greater than 1 or less?
Less than 1. So in this case, you'd have a larger number in your denominator than in your numerator. So as a fraction, it'd be less than 1. Yeah. So that's just something to keep in mind that I did not mention before. All right, so that brings us to the next part of our problem. So let's think about this reaction. 2A makes B plus C at 298K, same reaction, same concentrations, but now we have a different set of conditions. So now we're initiating our reaction with 10 millimolar of A, 20 millimolar of B, and 30 millimolars of C. So now we want to solve for delta G when the reaction is done at 298K. So does anybody know what equation we might want to use to solve this one? So we sort of have these non-standard conditions, or we're looking at a reaction at a particular point during its progress.
Correctly, I think it's delta G equals delta G naught plus RT log Q.
Yes. So delta G equals delta G naught plus RT times the natural log of Q. And does anybody remember the expression for Q? How would we find Q? Yeah. Yeah, exactly. It's kind of like K, but at a specific moment in time. So Q is still concentration of products over concentration of reactants. Okay. So where would we start with this? Or how would you start if you wanted to solve this problem? Yeah, finding Q is a good place to start. So do we have to do something before we find Q? Yeah. Yeah, excellent. So yeah, if it's ever given to you in millimolar, which you often will see millimolar or other concentrations in biological reactions, always convert first to molar. So first, let's get down our expression for Q. It will look a lot like our expression for K. So we have our products on top, concentration of B times concentration of C over concentration of A squared. So can someone tell me the concentration of B in molar? Yeah. Oh. Uh-huh. Okay.
If everything's given us, not given to us in millimoles, would we solve it in millimoles and then try to first solve it in moles?
I don't know if that would work because you'd end up with a unitless number, so there's nothing to really convert after. I've never tried doing it that way, so I can't tell you for sure it won't work, but I know for sure it does work if you change it to molar first, so that's what I recommend. So can anybody tell me what the concentration of B and C for our numerator, what would those be in molar? Or how would we convert millimolar to molar? Are you raising your hand or are you just looking? Okay, yeah. Yeah, exactly. So if we have, I'll just do A as an example. So if we have A equals 10 millimolar, so millimolar is equivalent to millimoles per liter. We want to change that to moles per liter. So we want to cancel out our moles, our millimoles, right? So we can do millimole in the denominator, mole in the numerator. We know that there's 10 to the third, 1,000 millimoles in 1 mole. Now our millimoles will cancel out. We're going to get an answer in moles per liter. It's 10 divided by 1,000. Divide by 1,000, we move the decimal 3 places to the left. So we get 0.01 molar, which is moles per liter. Does that make sense to everyone? Okay, so for B, we have 20 divided by 1,000, so we move the decimal 3 places over, 0.02 molar. That's our concentration of B. Our concentration of C, so we have 30 millimolar. Divided by 1,000, we have 0.03 molar. And then in the bottom, for our A starts at 10 millimolar. That's what we use for the example, so we have 0.01 molar. and that's squared because you have 2a in your equation. Does that make sense to everyone how we got here? Okay. So does anybody have a number for what Q is? Yes, Q is 6. So I'm just going to move over to the next slide because I ran out of space on this slide. So the next slide is just because I figured we'd all run out of space. So Q is 6. So that's great. Let's rewrite our equation that we're solving. So delta G equals delta G naught plus RT times the natural log of Q. So we know one part, we know Q. We also know R, right? R is our constant. R equals 8.314 joules per mole Kelvin. And since we're finding delta G, it makes our lives easier if we convert from joules per mole Kelvin to kilojoules. So that same idea, 8.314 joules per mole Kelvin. We want to change our joules to kilojoules. There's 1,000 joules in 1 kilojoule. Our joules cancel out. So we divide by 1,000 by moving this decimal place over. 0.008314 kilojoule per mole Kelvin. So that's our R. Does that make sense to everyone? And so what else do we need to know to solve this? We need to know T. So in our question, we know that T is 298K. So throw that over here. And the last, or the other, and we're solving for delta G. So we have R, we have T, we have Q. Do we have delta G naught? Did we solve for that a couple slides ago? Does anybody remember what delta G naught is? Yes, great. So it's 7.1 kilojoules per mole. And we know that because we figured it out a few slides ago. So yeah, now we can just plug everything in. So we have delta G equals delta G naught. So that's 7.1 kilojoule per mole plus R, which we already converted into kilojoule per mole Kelvin. So we have 0.008314 kilojoules per mole Kelvin. T, we know, is 298K. And then we have the natural log of Q, which is 6. So now our degree Kelvin cancels, so our answer is going to be in kilojoules per mole. So does anybody have delta G? 11.5, great. Okay, so what happened here? So we started out with our sort of standard conditions. Our delta G was 7.1. Is that spontaneous or not spontaneous? Not spontaneous. And then we changed around our conditions a little bit by changing our concentrations of our reactants and products. Now we have a delta G. Is that more positive or less positive? Yeah, it's more positive, so it became less spontaneous. So why do we think that happened? What did we do that made it less spontaneous?
Like the different concentrations of reactants and comets?
Yeah, exactly. So this reaction is not spontaneous. So at standard conditions, you have equal amounts of everything, right? And now we made these non-standard conditions. So we put in, or sorry, we didn't have equal amounts. We started out with this, right? That's where we are at equilibrium. So we changed it, right? So now for B and C, we changed B from sorry, I'm confusing myself. Okay, so yeah, okay, no, let me start over. So at standard conditions, your reaction is equal amounts of everything. That's part of what being at standard conditions means. So what we did was we changed that. We added not equal amounts. So we have 10 millimolars of A. That's less reactant compared to product, right? We have more B and more C compared to A. So if you add extra product to a non-spontaneous reaction, in a way you're trying to push it towards products, and it really wants to go the other way. And so it's it becomes sort of more non-spontaneous because you're not helping it out. Like, it wants to have more reactants, and instead you're giving it extra products. And so it's even less spontaneous, if that kind of makes sense. Yeah.
I'm a little confused about how at standard conditions,
Yeah.
having the same concentrations aren't the concentrations that you start off with also different for A, B, and C?
So yeah, I should have explained that. So this is at equilibrium. So you start out with equal amounts of everything, and then the reaction, because of delta G and delta H and delta S, it's going to go somewhere until it reaches equilibrium. And then when it reaches equilibrium, it's done. So like that's where we're at when we've reached equilibrium.
So there's one of the conditions for standard conditions, having one molar of everything?
Yes. Yeah. Mm-hmm. Yeah.
How strict should we be when it comes to significant?
Yeah, that is a good question. So when you're doing calculations, it kind of helps to carry along a couple of extra digits if you're going to kind of use that number for further calculations. But in general, you can sort of stick to the standard conventions of when you're given a certain number of sig figs, that sort of dictates how many are going to be in your final answer. For the purposes of quizzes and exams in this class, I will tell you how many sig figs I want. So in your quiz number 1, there's a few questions, a little bit like what we practiced today. And all of those questions, I ask you, give me 3 sig figs, give me 4 sig figs, give me 2 sig figs. So you shouldn't have to stress about that too much for the purposes of this class. Uh-huh.
So when a number becomes more negative, it definitely becomes more negative, we're allowed to say like the term it becomes more spontaneous, right?
Yes.
So what do you say like the reaction goes faster?
Right. Yeah, because faster relates to the rate, the kinetics. The spontaneity refers to the dynamics, not the kinetics. Any other questions? Okay, so if no one has any questions, I just want to give you some more information for next time. So next week we're going to start working on Chapter 2. I will put up the Chapter 2 lectures hopefully tonight, by latest tomorrow. And I will also, in addition to the lectures, I'll post the slides and that document with practice questions for you to work on after you watch the slides, or after you watch the videos, I'm sorry. And then similar to what we did today, next Thursday, we will go over some practice problems that are part of the slide deck. So that's what you need to know for next Thursday. Next Monday is Labor Day, so we're not meeting on Monday. And the other thing I'm going to do for Chapter 2 is we're going to have a case study. So I'm going to post a document with the case study and with some instructions on which questions from the case study. I believe it's questions 1 through 3 that you're going to be responsible for knowing, but I recommend that you go through the whole thing just for your own knowledge and learning. And the other thing that you'll need to know for next week is quiz number 1 is posted on Brightspace. It's going to open up right after class today, about 2:30. And you have until next Thursday, the next time we meet, to complete the quiz. It's all on Brightspace. You can use a calculator. You cannot work with a partner in a group. Yeah.
Quiz 1 is going to be for chapter 2?
Chapter 1. So on what we just covered. Yes.
Okay.
Yeah, so the quizzes are always going to be on stuff that we've covered and discussed, and then you will be quizzed on it.
Okay.
And then I'm going to always be posting the lectures. In the future, I'm going to get the lectures posted more ahead of time so you have more time to prepare, but I will never ask you to do a quiz on something we haven't already reviewed in class. Yeah, so keep your eye on Brightspace. Go ahead.
What's the format of quizzes? Is it like fill in the with your answer, or is there a chance for to figure out if you made math errors and is there any multiple choice?
So there's a mixture. So yeah, for the math questions, a lot of them are fill in. I think at least one of them I made multiple choice. Maybe not for this quiz because it's already posted, but in the future what I can do is add an extra space where you can show your work, and that way you can get partial credit. If it becomes an issue for this quiz, I'll maybe contact the students individually and ask them to send me their work just because it's not in the quiz and it's already up. Yeah.
Are they timed?
Yes, the quizzes are 30 minutes. There's 10 questions. Any other questions? Yeah. It's due next Thursday, so a week from today. I think I made it right before class starts. But I can make it Thursday night if you need the time. But yeah, maybe in the future I'll make it midnight. Any other questions? All right, so it's a little bit early, but that is everything we're sort of ready to do today. So yeah, so for next week, just keep your eye on Brightspace, do the quiz, and be ready to talk about Chapter 2. Have a great long weekend.